Hello guys, welcome to Dr. Science. So today we are going to talk about gram-positive lab algorithm. I know many of the second year students are studying microbiology and they are in a lot of difficulty in understanding this one. So I thought of making a video about it. And in this topic, I'll be mainly talking about gram-positive cocci. Okay, because many of the questions come from these are organisms. So first, you have to understand that what is meant by gram-positive. So gram positive means, suppose this is a bacteria, and when you do a gram staining, they appear purple in color. So this bacteria is appearing purple in color, right? So these are the organisms which are gram positive. Okay, and there is a mnemonic: all cocci are gram positive. Now, what is meant by this? So cocci means the bacteria which are in circle, or you can say round. These cocci are gram positive. All cocci are gram positive. It means all Staphylococcus, Streptococcus, they are round, right? They are circle or they are round. That's why they are called as gram positive organisms, and they are cocci. Okay. So there are mainly two exceptions which are cocci but not gram positive, they are gram negative, and that one is Neisseria and Moraxel. Okay, you have to remember these two organisms because they are cocci. I already said all the cocci here are gram positive. So Neisseria and Moraxella are cocci but they are gram negative. Okay, that is the important thing to understand. Okay, so I'll draw the diagram below, and after that I will explain this table to you. I'll be mainly talking about cocci, not bacilli and branching filaments. I will cover that one in different video. Okay, so suppose here are the cocci. Let me change the diameter. Okay, suppose these are the cocci organisms. What are these? These are the cocci bacteria. This cocci bacteria is divided into two types, mainly Streptococcus and Staphylococcus. What is meant by Streptococcus? Strepto means they are arranged in a chain. Okay, so coccus means they are circle or round in shape. So these bacteria are arranged in a chain. Okay, these are arranged in a chain, that's why they are called as strepto. And coccus means they are in circle, right, or round in shape, that's why they are called as coccus. And what is meant by staphylococcus? Staphylo means they are arranged one above the other. So these are the cocci organisms which are round in shape and they are arranged one above the other, like this. That's why they are staphylo. Staphylo means one above the other. So they are arranged into the gram positive cocci bacteria. Gram positive cocci bacteria are arranged into Streptococcus and Staphylococcus. But based on what? Based on what they are arranged in Strepto and Staphylo. So you have to understand that Staphylococcus is mainly a catalase positive. Staphylococcus is catalase positive, whereas Streptococcus is catalase negative. But what is meant by catalase? What is catalase? So catalase is an enzyme. Okay, so here is your cat. Imagine this is a cat and it have a knife here. Okay, so catalase is an enzyme which is present in your bacteria. Enzyme present in your bacteria. And what does this catalase do? Catalase, normally hydrogen peroxide, is a very toxic substance, as you know. Normally this toxic substance is present in our cells in order to kill the bacteria in our cells. Okay, in humans mainly, in humans this hydrogen peroxide is present, and what we this hydrogen peroxide can kill the bacteria. So what does the bacteria do? This bacteria produce an enzyme called as catalase. So here is our cat, and this cat is having a big knife, and with this knife it will cut down the hydrogen peroxide into mainly water and oxygen. Okay, so that is a function of this catalase enzyme. So what we are getting when you are doing a catalase test, when do you suppose this is the slide, and on this slide we are putting the bacteria. Okay, so what will happen if the bacteria is having a catalase? Obviously the H2O2 will convert into water and oxygen. So whenever you see oxygen in water, what will happen? Yes, suppose this is a test tube and there is water in it, and if there is oxygen you will form a lot of bubbles, right? Similarly on the slide test you will form a lot of bubbles, air bubbles, because of oxygen, right? When oxygen air bubbles are found on the slide test, oxygen air bubbles are forming on the slide test, it is known as catalase positive organisms. Okay, I hope you understand this concept. Okay, so based on catalase positive test, it means Staphylococcus will produce oxygen on the slide test. Okay, whereas Streptococcus doesn't produce oxygen, no oxygen. That's why Streptococcus is a catalase negative bacteria, whereas Staphylococcus is catalase positive bacteria. So again we will draw the diagram. So here is cocci, gram positive cocci, and based on catalase test, we are dividing it into catalase positive organisms, which are Staphylococcus, and catalase negative organisms, which are Streptococcus. Okay, now we will talk about Staphylococcus, because Staphylococcus we found a lot of organisms which are very important and we have to understand them. So Staphylococcus is a gram positive and it is anaerobic. Okay, all these cocci we are talking about are facultative anaerobes. Anaerobic, what is meant by anaerobic? First you have to understand the difference between aerobic and anaerobic. Aerobic means they love oxygen. Okay, anaerobes means they don't love oxygen. But what is meant by facultative anaerobes? Facultative means they can work in the presence or absence of oxygen. It is both in the presence or absence of oxygen. Okay, so Staphylococcus is again divided into two categories, and in those two categories we will be talking about Staphylococcus aureus and Staphylococcus aureus, and the other two are Staphylococcus epidermidis and Staphylococcus saprophyticus. Okay, so first we have to understand that what is Staphylococcus. It is a gram positive organism. It is an anaerobe, right? And the second thing was, it is what, catalase positive, right? It is catalase positive. So how are we dividing this Staphylococcus again into Staphylococcus aureus and Staphylococcus saprophyticus and epidermidis? Actually, we are dividing the Staphylococcus into aureus and saprophyticus based on coagulase enzyme. And coagulase positive is Staphylococcus aureus, coagulase negative is these two organisms. Okay. So what is this coagulase enzyme? You said catalase was an enzyme which is producing oxygen, right? Catalase will produce oxygen. But what is coagulase? What is this coagulase? We don't know. We have to differentiate these two, okay? Coagulase and catalase. Catalase is an enzyme which will produce oxygen and on slide test. But what is this coagulase? So suppose this is a bacteria. Bacteria is having this coagulase enzyme. The function of this coagulase enzyme is, actually there are two types mainly, free and bound coagulase. What is meant by free and bound coagulase? Suppose this is the bacteria. On this bacteria cell wall, there is coagulase enzyme attached on it. This is called as the bound coagulase. And there is coagulase which is free inside, okay? This is called as the free coagulase. There is bound coagulase and there is free coagulase in Staphylococcus. So mainly we will be talking about, for this video I'll be mainly talking about coagulase enzyme, because bound and free coagulase, they are a different topic and I will cover it in a later video. But we have to know the function of this coagulase enzyme. So suppose this is a bacteria and this bacteria is having coagulase enzyme. The function of this coagulase enzyme is for the formation of coagulation. So what this coagulase do? Normally, suppose this is a human cell. This is the human cell, and not human cells. Suppose these are the humans, and normally humans in their body have fibrinogen. Okay, this is a fibrinogen is a clotting factor, right, which is present in the plasma of the humans. So what happens? The bacteria have coagulase enzyme. This is the coagulase enzyme, and the humans have, yes, it is fibrinogen. So suppose this is the fibrinogen and this is the coagulase. They both interact with each other and they both activate each other, and together they will form a clot. Okay, so if you take a slide test, on a slide test, if a bacteria is having coagulase enzyme, obviously on the slide you will form a lot of clots, right? There is a lot of clots formed because the bacteria is having a coagulase enzyme, and on this slide we are putting the plasma of the humans. In plasma we are having fibrinogen, so both of them will fuse and they will form multiple clots. Okay, So that was about coagulase. So what is the function of coagulase? Coagulase will cause clots on the slide. So coagulase positive is Staph aureus. I already told you Staph aureus is coagulase positive, and coagulase, actually they are called as COPS. COPS stand for coagulase positive Staphylococcus aureus. Coagulase positive Staphylococcus aureus. Okay. Whereas cons, CONS, cons stand for coagulase negative Staphylococcus, which are Saprophyticus and Epidermidis. Okay. So we will take a look at the diagram again. So I will draw here again so there will be no confusion. So there are gram positive organisms, and we are mainly talking about cocci. So this cocci is again divided into Streptococcus and Staphylococcus. Based on what? Based on catalase positivity. Catalase positive is Staphylococcus, catalase negative is Streptococcus. Staphylococcus is again divided into two types based on coagulase enzyme, right? Coagulase positive is Staphylococcus aureus. It means Staphylococcus aureus will form clots on slide, whereas coagulase negative are Staphylococcus saprophyticus and Staphylococcus epidermidis, which are coagulase negative because they will not form the clots on the slide. I hope you understand this concept, which is very important. So I want to go a little deeper because nowadays they are not asking superficial topics, they are going a little deeper. Again, the Staphylococcus aureus, I mean Staphylococcus is divided into Staphylococcus aureus based on coagulase positivity. Okay, so this is coagulase positive organism, whereas Staphylococcus saprophyticus and epidermidis is again divided by a test. Okay, I know this is a lot deeper for second year students, but trust me, they are asking a lot of questions on this one. So Staphylococcus saprophyticus and Staphylococcus epidermidis. But on what basis? Obviously, this is coagulase negative. Both the saprophyticus and Staphylococcus epidermidis, they are coagulase negative, and they will not form a lot of clots on slide test. Okay, but on what basis they are differentiated into two organisms? Yes, there is a sensitivity test, that is novobiocin. Novobiocin is like an antibiotic, okay, which will kill the bacteria. So suppose we have taken two organisms on a slide test, not a slide test, I'm just generally speaking, and these two organisms are, this organism is Staphylococcus saprophyticus and this organism is Staphylococcus epidermidis. So what is happening in this novobiocin sensitivity test? As I already told you, novobiocin is an antibacterial. Antibacterial means it will kill the bacteria. And no, if we add a tincture of, like, if we add novobiocin on this organism, which is an antibiotic, it will kill the Staphylococcus epidermidis. Okay, now the Staphylococcus epidermidis is completely dead because we added an antibacterial which will kill the bacteria, which is Staphylococcus epidermidis. It means it will kill the Staphylococcus epidermidis. That's why it is novobiocin sensitive. Okay, Staphylococcus epidermidis is getting killed by the novobiocin antibiotic. That's why it is called as novobiocin sensitive positive test. Okay, whereas Staphylococcus saprophyticus, novobiocin can't kill that organism because of resistance or some other thing. That's why it is novobiocin negative sensitive, not sensitive to novobiocin. Okay, so I hope you understand that concept. Again I will explain. So Staphylococcus is again divided into Staphylococcus aureus. I will not go that deep. Let's uh, differentiate Staphylococcus saprophyticus from Staphylococcus epidermidis. So based on novobiocin sensitivity test, novobiocin sensitive is positive for Staphylococcus epidermidis, which will kill the organism, whereas novobiocin will not work on Staphylococcus saprophyticus. That's why it is novobiocin negative cell. Okay. Okay. Now let's go to a different topic, which is Streptococcus. So again we will draw this one, gram positive cocci, which are divided into Staphylococcus and Streptococcus based on Catalase test. And in Catalase positive was Staphylococcus, Catalase negative was Streptococcus. And now we will talk about the Streptococcus, which is gram positive, Catalase negative. Okay, this is the important word here. So this Streptococcus is again divided into, okay, before continuing to the next topic, I will just show the diagram so your basics will get good. So I already told you gram positive are purple organisms and they are cocci, they are anaerobic and facultative. Based on catalase testing, catalase positive organisms are Staphylococcus, and this Staphylococcus is again divided based on coagulase positive or negative. Coagulase positive is Staphylococcus aureus, which will form a lot of clots on the slide. Okay, whereas coagulase negative is the Staphylococcus saprophyticus and epidermidis, right? Coagulase, these COPS are the Staphylococcus aureus, whereas CONS, coagulase negative Staphylococcus, are the Staphylococcus saprophyticus and epidermidis. Again they are divided based on novobiocin sensitivity, whereas novobiocin sensitivity will kill the Staphylococcus epidermidis. That's why it is positive, whereas novobiocin saprophyticus will not kill saprophyticus. That's why novobiocin is negative for sapro. Okay, now I will not discuss this. I will discuss the next step is Streptococcus, which is a very important topic. And I understand that you know about the Landsfield classification, because that one is very important. So many questions have been asked in the past also. So I will not go into the deep, but Dr. Lansfield, what he did was based on the cell wall carbohydrate. You have to remember this one. Okay, the cell wall carbohydrate, or also known as the aka C carbohydrate. C carbohydrate divided the Streptococcus organisms into many categories or groups. And you have they previously they asked what is the classification, Lansfield classification, based on, is it carbohydrates, fats, lipids, or something like that. The answer was C carbohydrates. I hope you guys will learn this. So let's get into the Streptococcus topic. I'll discuss in this one in the next video.